Okay. See, the spontaneous, that is continuous diffusion of water across a differentially or semi-permeable membrane, we call it as osmosis. The movement of water from a higher uh, concentration to a lower concentration through a semi-permeable membrane. So you can also call it as, since we are using the term from higher concentration to lower concentration, so you can also use the term from higher water potential to lower water potential through a semi-permeable membrane. We can call it as osmosis. Like here we have the cell membrane, tonoplast. Tonoplast is the outermost membrane of this vacuole. They are important. So these two membranes, cell membrane and tonoplast, are important determinants of movement of molecules in or out of a plant. So two of them decide which molecules has to enter and which molecules has to exit. One is the cell membrane, another is the tonoplast, which is the outermost membrane of the vacuole. We call it as tonoplast. They decide which molecule has to enter and which molecule has to exit. Okay, but the cell wall is not a barrier to movement as it is permeable to water and substances in solution. So the cell wall is not a barrier for movement because they are going to be permeable to water and substances which are dissolved in water. They can enter through cell wall. The barriers are going to be cell membrane and tonoplast. So the cell membrane and tonoplast determine what substances has to enter and what substances has to exit. The entry and exit of molecules are determined by cell membrane and tonoplast. Okay, but cell wall is not a barrier because it is permeable to water and uh, solutes dissolved in water. That is solution. Okay, permeable to water and substances in solution. So the vacuolar sap in large central vacuole, you know that plant cell has a large central vacuole. So they have the sap within this vacuole. So vacuolar sap in large central vacuole, they contribute to the solute potential of the cell. So because this will be having solutes dissolved in them, so the vacuolar sap will be uh, in the central vacuole is going to determine the solute potential of the cell. So the vacuolar sap equals psi S. That is, they are the solute potential or osmotic potential of the cell. So here there is an osmosis that they have shown it through a different solution. See, solution, what is a solution? Define a solution. Can you tell me what is a solution? Define solution. Any one
When a
of you?
solute is added to solvent, it becomes solution.
Yes, the dissolution of solute in a solvent, we call it as solution. Solution equals solute plus solvent. When a solute is added to a solvent, they dissolve and they form the solution. What are the different types of solution? What are the different types of solution based on their concentration?
Hypertonic,
Heterogeneous.
hypertonic and isotonic.
Very good. Hypertonic, isotonic, hypotonic. Okay. So for how do you call any solution as hypertonic?
When it is having more concentration.
How do you know that it is having more concentration?
Exo- endosmosis.
See, you should compare two solutions. There should be a reference point. When two solutions are there and one of them is having a higher concentration than the other solution, then we call it as hypertonic. So with reference to cell sap, if a solution is having higher concentration, so we call it as hypertonic. With reference to the cell sap, that is the cytoplasm, if they are having same concentration, if a solution is having the same concentration as that of cell sap, we call it as the isotonic. If they are having a lower concentration than the cell sap, solution is having lower concentration than the cell sap, we call it as hypotonic. Okay, So water, pure water is hypotonic. There is no solute at all. Okay, or hypotonic, it can be lower concentration than that of the cell sap. So whenever we are defining hypertonic, hypotonic, isotonic, we do it with reference to cell sap or another solution we take up. In your chemistry lab, you're telling us one solution is hypertonic, you should be having another solution in comparison. Only in comparison you can say about hypertonic, hypotonic, and isotonic solutions. Okay, so that is one thing that you have to remember. So based on the strength of the solution, what are the different types of solutions that you can prepare based on their uh, strength? What are the different types of solutions? If molecular weight of a substance is dissolved in 1 liter of water, what do you call that solution as? What are the different types of solutions that on the basis of concentration, what do you have in your chemistry?
Concentrate and diluted.
So that is simple words. Okay, you have a molar solution. Do you have it or not? Okay, so in 1 liter of water, if molecular weight of a substance is dissolved in 1 liter of water, you call that as molar solutions. Normal solutions, normality solutions, percentage solutions based on the percentage of the solute dissolved in the water, you have percentage solution. So equivalent weight of a substance is dissolved in a solvent, you have another type of solutions. So different types of solutions based on their uh, strength of concentration is, so normal solutions, normality solutions, molarity solutions, percentage solutions. Okay, so remember about this different types of solutions on their strengths. Maybe they might not have given here, but this is a little bit of chemistry that you should be aware about. Now we will be taking up the solution A and solution B. In solution A, the number of solute molecules are less, so water potential will be higher, or the water concentration will be higher in solution A. Solution B, they'll have more of solute and water concentration will be lesser. That is, water potential is lesser. Solute potential is low, water potential is high. Solute potential is high and water potential is low. Okay, so solution A, it has high water potential, high solute potential. So solution B, it has low water potential, low solute potential. Okay, so as a result of that, so there is movement of water molecules from a region of higher water potential or higher energy, free energy, to a region of lower free energy through a semi-permeable membrane. It is taking place. So this is the direction of movement of water. So this we call it as osmosis. Osmosis is the movement of water from a region of its higher free energy to a region of its lower free energy through a semi-permeable membrane. We call it as osmosis. So the net direction and rate of osmosis depends on the difference in pressure, pressure gradient, and concentration gradient. So here the movement of water is based on concentration gradient. Sometimes differences in pressures also results in pressure gradient, and movement of water is because of pressure gradient. So the net direction and the rate of osmosis depends on two factors. One is pressure gradient, another is concentration gradient. So water moves from its region of higher chemical potential or concentration to its region of lower chemical potential until equilibrium is attained through a semi-permeable membrane. At equilibrium, the two chambers should have the same water potential. Only then you can say that they have attained equilibrium by osmosis process. So this you'll be doing in the lab, potato osmoscope we call it as. See, osmosis can be demonstrated by this potato. So what they have done is they have taken a fresh potato, they have peeled off the skin and cut it into a cup shape, and they have scooped off some potato tissues to make a cup-like structure. So the solution that we have here is it can be salt solution or sugar solution. you can have it. Initial level of this solution is marked by a dot pin. So you can initially mark the solution level by a dot pin. So leave it in a beaker containing water. Okay, so after a few hours, half an hour or 1 hour, you see that the level of solution would have raised. So mark it by another dot pin. So you see that the level of solution would have raised within this osmoscope. What is exactly happening here? See, the water, pure water is there in the beaker. It moves through the semi-permeable membrane and it enters into this cup-shaped structure. So since this is taking place through living cells, we call it as physiological process. It is a potato osmoscope, we call it as. So it is a physiological experiment. Since it involves the living cells, we call it as a physiological experiments. Okay, and you can demonstrate osmosis, which is occurring, the movement of water from a region of higher water potential to a region of lower water potential through a semi-permeable membrane. So the potato cells, cell walls and membranes, they are acting as semi-permeable membranes. So we call this as osmosis. So make a cavity in a potato tuber. So you scoop out those tissues. It is a fresh potato. The outermost skin is peeled. So in this, we are pouring concentrated sugar or salt solution. So this setup is called as potato osmometer. So this is placed in a beaker containing pure water. Okay, so place it in water. As a result, water enters the cavity due to osmosis. So this experiment to demonstrate osmosis using a living tissue, we call it as the physiological experiment, potato osmoscope experiment, or potato osmometer. So you can see before osmosis, the level of this dot pin was lower. After osmosis, the level of water within this cup is higher. So that is because of osmosis. So Thistle funnel experiment, we call this as the physical experiment. So you can notice in a Thistle funnel, it is inverted. The Thistle funnel is inverted. So we have covered the mouth of the Thistle funnel with a semi-permeable membrane. Okay, it can be the membrane of a goat or cheesecloth. So the mouth of this funnel is covered with this semi-permeable membrane, which can be a membrane from goat or eggshell membrane, or it can also be a any other semi-permeable membrane. So we have tied the mouth of it, and within the Thistle funnel we have this concentrated solution, and this is placed inverted into the beaker. Okay, so there is the entry of water from the beaker, it enters into the Thistle funnel due to osmosis. And here there are no living tissues involved. All of them are dead. Even this membrane is not living. So it is just semi-permeable and it is dead tissue. So what do you notice is there is a movement of water even in a physical setup. So, so osmosis is a physical process. You cannot say that it can take place only in living system. So osmosis takes place in a living system as well as in a physical setup. In a non-living atmosphere also, the osmosis takes place. So the inverted Thistle funnel experiment, we call it as, which is also called as physical experiment. Physiological experiment is potato osmoscope, or we also call it as potato osmometer. So in the physical experiment, what we are doing is a Thistle funnel is filled with sucrose solution. It is kept inverted in a beaker which is containing pure water. So the sucrose solution is separated from the pure water by a semi-permeable membrane. So the mouth of the Thistle funnel is having a semi-permeable membrane, especially the eggshell membrane. So water moves into the funnel. As a result, the level of the solution in the funnel rises. It continues till equilibrium is reached. So that is in both water and solute, they should have the same concentration. So same water potential. When they reach that, equilibrium is attained. Now, if the second experiment in the B, what they have done is in the Thistle funnel, they have applied pressure. See, this pressure that we have applied prevents the osmosis from taking place. Okay, so if an external pressure is applied from the upper part of the funnel, no water diffuses into the funnel through the membrane. So this pressure required to prevent water diffusion is called as osmotic pressure. So the amount of pressure that should be applied to prevent water osmosis, the osmosis to happen or diffusion of water to take place, we call it as osmotic pressure. The pressure required to prevent water from diffusing, we call it as osmotic pressure. So this is a function of solute concentration. More the solute concentration, greater will be the pressure required to prevent water diffusion. Numerically, osmotic pressure is equivalent to the osmotic potential, but the sign is opposite. Osmotic pressure is positive and osmotic potential is negative. So now you can prevent the diffusion of water by applying pressure. So the pressure required to prevent the water from diffusing, we call that as osmotic pressure. Okay. This is also a function of solute concentration. More the solute that is present in the Thistle funnel, numerically osmotic pressure is equivalent to the osmotic potential, but their sign values are opposites. Osmotic pressure is positive and the osmotic potential or solute potential is negative values. Okay. So if an external solution balances the osmotic pressure of cytoplasm, it is called as isotonic. This is another good way of defining it. If there is an external solution which can balance the osmotic pressure of cytoplasm, we call it as isotonic. When a cell or tissue is placed in isotonic solution, there is no net flow of water either inside or outside. Water flow is in equilibrium. Such cells are said to be flaccid. See, if an external solution balances the osmotic pressure of cytoplasm, so that external pressure which balances the osmotic pressure of cytoplasm, that solution, we call it as isotonic. When a cell or tissue is placed in a isotonic solution, there is neither entry of water nor exit of water. There is no net flow of water towards inside or outside. That is, neither entry nor exit of water takes place. Then the cytoplasm, such cells where there is neither entry nor exit of water taking place, we call it as such cells are said to be flaccid. If the external solution is more dilute, higher water potential than the cytoplasm, it is hypotonic. Cells swell in hypotonic solution. So cells are going to swell up. वो थोड़ा cells whenever they are in hypotonic solution, cells swell because of entry of water in hypotonic solution. If external solution is more concentrated than cytoplasm, it is hypertonic. We are talking about hypertonic solution. The cells are going to become here, when a cell is placed in hypertonic solution, water moves from the cell across the membrane to outside. So the area of lower water potential. So from area of higher water potential, they move towards the area of lower water potential and the cell shrinks. So the shrinkage of cell, whenever it is placed in hypertonic solution, the shrinkage of cell, we call it as plasmolysis. Water is first lost from the cytoplasm and then from the vacuole the water is lost. So during plasmolysis, the cell membrane and protoplast of a cell wall of a plant cell shrink away from its cell wall. So you can notice that the cell membrane and the protoplast of a plant cell, they shrink away from the cell wall. So such cells are said to be plasmolyzed cells. You'll be doing an experiment in lab where you have to see this, demonstrate this plasmolysis. A normal cell, when it is placed in a hypertonic solution, they lose water from it because of difference in water potentials. So there is a loss of water and shrinkage of cell takes place. Okay. So the shrinkage of the cell, we call it as plasmolysis. This process is plasmolysis. So during plasmolysis, the plasma membrane and the cytoplasm or the protoplast of a cell, they shrink away from cell wall. Such sort of cell where cytoplasm is shrunk, where the plasma membrane is shrunk, we call it as plasmolyzed cell. So this is before plasmolysis. After plasmolysis, this is how you see it in your microscopes. So plasmolysis is reversible. How can you reverse this plasmolysis? So place it in pure water or hypotonic solution. When you place a plasmolysed cell in pure water or hypotonic solution, so they become normal cells. So plasmolysis is usually reversible. So we can deplasmolyze this by placing it in a hypotonic solution or pure water. So as a result, the cytoplasm builds up a pressure against the wall. It is called as Turgor pressure. Okay. Whenever cells are placed in a hypotonic solution, we have already told you it swells up. As a result, the cytoplasm builds up a pressure against the wall. It is called as Turgor pressure. The pressure exerted by the protoplasm due to entry of water against the rigid walls is called as pressure potential, psi p. The cell does not rupture due to the rigidity of cell wall. So Turgor pressure causes enlargement and extension growth of cells. So Turgor pressure causes enlargement and extension growth of cells. Then the next concept that we have to discuss is imbibition. It is a type of diffusion in which water is absorbed by solid hydrophilic colloids, increasing their volume. So initially it would be adsorption. They have not mentioned here, but it is adsorption of water followed by absorption of water. So by hydrophilic colloidal substances. So imbibition is also a type of diffusion in which water is adsorbed by solids, that is hydrophilic colloidal substances, increasing their volume. So the absorption of water by seeds and dry wood, we call it as imbibition. It is because of imbibition. Okay. The pressure due to the swelling of wood can split rocks, which I have told you in the previous class. The seedlings are emerged out of the soil. The germination of seeds is because of imbibition pressure. So imbibition requires difference in concentration gradient, water potential gradient between the absorbent and the liquid imbibed. That water potential differences should be there for imbibition to require. So the affinity between adsorbent and liquid, that interrelationship or similarities between adsorbent and liquid should also be there. So they should have closeness between the adsorption and the liquid is also going to facilitate imbibition. So imbibition requires various parameters are there for imbibition to occur. They should be difference in concentration gradient, water potential gradient between the adsorbent and the liquid imbibe should also be there. So there should be a difference in water potential, and affinity between the adsorbent and the liquid should also be there. So these are the parameters required for imbibition to occur. So next part that we'll discuss is the long distance transport of waters. Okay. I think we'll start this with the next class of it, long distance transport of water, because it is a continuous concept. So we'll just have a revision of what we did for today. Okay.